Do we consider viruses to be living as a biologist? No, right? Because it depends on what you consider to be living, right? And from a biology perspective, what deems something to be living, they have to fit 7 different criteria, right? You have to be able to do reproduction, right? Homeostasis, response to environment, right? All that cool jazz. So since it doesn't fit in to what is living, it doesn't fit into our they, right? Bacteria, archaea, eukarya. It's its own separate thing. So even when we talk about how we name viruses, they don't get the same binomial nomenclature. So they don't get genus species. We name them a little bit differently. Okay. Yes, that's all I had to say about living. Okay. How did we first discover viruses? We first found viruses in actually a plant. Okay. So back in 1886, this guy Adolf Meyer, he found that TMD, that's tobacco mosaic disease. This is a disease that was affecting tobacco plants. Okay, why do they care? Back in those days, tobacco was a really huge good, right? That's how they made a lot of their money. So if there is a disease that's attacking their crops, that's going to be a problem to them, right? So they're going to investigate it. So, but it wasn't until 1982 that they found that this disease that was infecting plants was something that was filterable. What they mean by filterable means that at this point, we knew bacteria existed. So we knew bacteria were a thing. We didn't know viruses were a thing. So whenever they extracted a diseased plant and they would place the material through a filter paper, whatever went through the paper was able to still infect a healthy plant. So that means that whatever's causing the disease doesn't get stuck in the filter paper. It has to be smaller than that filter paper, if that makes sense. So we know whatever we have is smaller than bacteria. Okay, that was the thought process.
So filterable means that it can go through the filter?
It goes through the filter.
So not filterable means that it gets stuck on the filter?
Yes. So the bacteria would be not filterable, but this guy was going straight through the filter, so it had to be smaller. Then 1930s, that's when they came up with the word virus, which they got derived from, I guess, poison, and they used that term to describe the disease, right? So whatever this disease was poisonous to the plant, right? It's killing the plant. And then 1935, that's when Wendell Stanley actually isolated tobacco mosaic, the tobacco virus, which caused tobacco mosaic disease. So again, 1935, that's when we had the electron microscope. So now we could actually ID the virus. Okay. So the very first virus we discovered wasn't even a virus that infects us. It was from a plant. Pretty cool.
Wait, bacteria are not
Just think about it like
in this scenario. So right here, we're trying to figure out what is causing disease in this plant, right? And we didn't have an electron microscope. So we couldn't see a virus. We didn't know what a virus was. We did know about bacteria. So they designed like a filter paper, kind of like when you filter out your coffee and the grains stay at the top. So the way that they made the filter paper was bacteria could stay stuck at the top like your coffee grains. So when they filtered it and they collected the material at the bottom and they used that on a healthy plant, the healthy plant would then get the disease. So they're like, whatever is causing disease can go straight through this filter paper, AKA smaller than bacteria. And that was the only thought process.
in this term filterable going through.
Goes through the filter.
But if we were talking in terms of bacteria would be filterable even like
No, it wouldn't be filterable means goes through the filter.
Just think about like when we filter water, water is filtered now. Because it goes through the filter.
okay.
here's my talk, right? What is living, what is non-living? So viruses have to use a host cell in order to replicate, whether that's a plant cell or an animal cell or fungal cell, right? It has to be inside of a cell in order to replicate, okay? Viruses in their most basic form, not that they're not able to be infectious, right? We have certain viruses that can stay on like inanimate objects and still cause infection. However, for them to reproduce or replicate, they have to be inside of a cell. So that's like our big, absolutely not, right? Yes. Okay. A virus in its most basic form has to have genetic material, okay? Some viruses are DNA, some viruses are RNA, okay? But you can't have both. So you don't have a virus that's DNA and RNA. You either are a DNA virus or you're an RNA virus, okay? We'll talk about both types. If you're an RNA virus, they actually get like even fancier. They're like single-stranded RNA, double-stranded RNA. They're like positive single-stranded, negative single-stranded. We'll get to that later. But all viruses have to have some sort of genetic material in its just most basic form. They also have to have some sort of protein coat. Okay, the protein coat is going to protect that DNA material. Okay. When a virus replicates inside of a cell, those new viruses that you just made, we call them virions, aka baby viruses. Okay? When a virus replicates and makes a bunch of viruses, the little like baby viruses, they just get a fancy word, virions. Oh, those virions, right? We'll use that word later. It'll sound all fancy, right?
Sounds expensive.
Huh?
They sound expensive.
Right, like, oh yeah, these the virions, like, I don't know why it goes into a different accent, but, yeah, anyways, right? Viruses, they're basically a parasite, kind of, right? They're kind of a parasite because what do they have to do? They have to go inside of another cell. So a virus, in its nature, is a parasite. Right? Because you just go back to what the definition of parasite is, right? And they get fancy and they say it's an obligate intracellular parasite. Okay? There's a few obligate intracellular parasites. That would be something like malaria, right? Malaria is an obligate intracellular parasite because it has to go inside your cell. Toxoplasma is an obligate intracellular parasite. All viruses have to be intracellular though, so that's kind of like redundant. But you can use that term to describe their behavior, right? Obligate means they have to, intracellular has to be inside the cells, and it's parasitic in nature, right? So it's kind of fun to say, like, oh, the virus is an obligate intracellular parasite. I think it's fine. Okay, so for size reference, okay, we have E. coli, right? And it's peritrichus, right? We learned that already, right? It has a bunch of flagella all over, right? Peritrichus. And then look at the virus, right? Look at all these different viruses. Super tiny compared to your E. coli, right? This one is even smaller than your ribosome, okay? Viruses do not have ribosomes. Some of you will get that wrong. You'll be like, yeah, viruses have ribosomes, sounds great. And you like circle it with like all this confidence. Remember, viruses don't have ribosomes, okay? Some viruses do come with their own proteins that help them like take over, but not all of them do. But some of them are a little fancy and they come with their own proteins. But if you look at these guys, this is what's called a naked virus. Ooh, naked, right? Because it's just genetic material and a protein coat. This one, herpes virus. This one, you have DNA, your protein coat, and then you also have a membrane, okay? If a virus has a membrane, then it's called an enveloped virus, right? The membrane is the envelope, okay? When you think of in terms of susceptibility, a enveloped virus is more susceptible than a naked virus. Any thoughts why?
Because you can attack the envelope easily.
Yes, you can attack the envelope easily, right? So just like Dawn, right? Dawn will do it, right? So like COVID is an enveloped virus. So if you wash your hands with Dawn, you can get rid of that envelope, right? So that's pretty cool. Naked, a little harder to target. This envelope can also have spikes on it. Okay, these spikes are different proteins, and that's also going to help. It helps the virus actually get into your cell, and we'll talk about that. But for now, they can have different shapes. Okay, so like this guy, bacteriophage, kind of looks like a little alien. Okay, this one's a polyhedral, so like it has many sides. Okay. Poxyvirus is weird. I'm going to tell you all the things that how a virus should be made, and then poxyvirus is just like different. Like I'd be like, star, this one doesn't fit everything that we're talking about. That one's just like our exception, right? We always have an exception to the rule. Is this one? It's the poxyvirus. Yeah, that one is weird. It doesn't fit everything that we say about the viruses that we're going over, right? And then for reference too, okay, here's a human liver cell, way bigger than E. coli. And then look at your virus, right? We're talking about nanometers. So super, super tiny. Okay, so in black is what all viruses have, and then in red may vary, okay? So a virus, right, or a virion, if you want to be fancy, a virion has a central core, which is going to be made of the genetic material, right? That genetic material could be DNA, it could be RNA, okay? What they can also have are matrix proteins. So these are these enzymes that it brings with it, and it's either going to help in taking over cell machinery, it's going to help it some way in replication, or it's going to help it some way to exit the cell, okay? So sometimes it brings in like its own tool, okay? But these tools alone will not help your virus replicate by itself, okay? We still need the host cell. Then you have a covering, right? Your covering is going to be a capsid. Capsid is your protein coat, okay? Capsid is your protein coat. Yeah. Some can have an envelope, okay? Not all viruses have envelopes. So here, in purple, this capsid, the capsid is the covering, and the capsid is made up of capsomeres. So if you think about these tiny little circles, those are the capsomeres, and together they make the capsid, okay? But it's all protein, okay? And it looks like this one, its core is DNA, okay? This one has an envelope, and it has spikes, okay? So basic structure of a virus, okay? Your capsid can come in different shapes, okay? Their purpose is to protect the DNA or RNA, right? Protect the nucleic acid. Each tiny subunit, so each little ball right here, would be a capsomere, and together the structure is the capsid. This is a polyhedral virus, so poly means many. So it's if you look at, like, this is a triangle, this is a triangle, This is a triangle. So it has many sides, right? The capsid is useful when it comes to detecting a virus, so we can do like PCR or antibody testing on through the capsid in order to identify what virus you have. Okay, so that's kind of cool. And the capsid is what's going to attach to your cell in order to enter your cell. Questions? Okay, so some viruses, again, are naked. Naked one's the one at the top, and then envelope is at the bottom. Okay, so sometimes you can have spikes. If you do have a spike, it's typically a carbohydrate protein complex. So it's some sort of protein / carb together. Okay, these guys are also useful in identification, right? So we can also use these for like PCR testing or antibody testing, right? That's how we test for COVID, right? We test for these spikes, and that identifies if you have it, yes, or no, you don't have it. Okay? These guys also help when it's when coming into the cell. It kind of sucks because in order to get a virus, you have to have a receptor on your cell that accepts this virus. It's kind of ugly, right? Because, right, receptors, ligand interaction, it's like a locked key mechanism. So the only way that this virus gets inside you is your receptor on your cell said, hello, come on in. It's kind of neat, right? So your own cells are accepting it. That's kind of crazy to me, right? Your own cells are accepting it.
So like active sites or outside sites?
It's a receptor. So like your receptor will recognize that spike and then it can come inside. And that goes a little bit into a little bit of genetics. So for example, some people, not that they can't get COVID, but some people are more prone to COVID, and that means that you have way more receptors on your cells than somebody else. Why do you have way more receptors? Genetics. Blame your parents. Yes?
We went to like this thing for extra credit with the couple semesters ago, and they were talking about like something like this with HIV, but I guess like there's a mutation amongst I think German people, and they don't have the receptor, so it's harder for them to get HIV or they can't get it.
They won't get it.
Yeah.
Because HIV is also an enveloped virus with spikes on it, and there is a group of people that they have a mutation where their receptors do not recognize the HIV spikes, so they cannot get the HIV virus. Well, the disease, right? If the virus comes inside, they don't recognize, so if it can't come inside and replicate, you're not gonna get no disease. There's also a level where they have some, like from one parent, they got the mutation and the other parent, they don't. So they actually have a lower risk level of getting it, but they do have the ones, like if you have both of the genes with the mutation, then you don't get it at all. It's a very small percentage, but that's crazy, right? So you can blame your parents. If you need another reason to be mad at them for something, be like, you did this to me, right? If you always get like colds all the time, there you go. Be mad at your mom or dad. You did this to me. It was your genes. Yes? One, they are new to a lot of things, so they haven't built their immunity. Two, a lot of things can be prevented just by like hygiene. And kids don't have the best hygiene, right? They touch the floor, they put their fingers in their mouth. So one, it's mainly like hygiene. And then two, they don't have their library bank of microbes that they've seen that they can quickly have antibodies to. Once you get older, you already have antibodies for different things. So when your body sees it, boom, takes care of it right away. Versus kids.
I read an article where like in certain populations they've significantly dropped like allergies to like peanuts or soy because they've exposed kids in those populations to it very, very early.
Yeah.
So then they were not allergic to it at all.
Exposure therapies, that works. Because if you never, that's why like, it's not good to be like in a bubble and like don't touch anything, don't put like, because then you're never building a library, right? If you're never building a library of defenses, then anything can knock you down. And that actually happened with like polio. When we talk about polio, we eradicated polio so much that our body didn't know what to do when it came into contact with polio, and then we got like super sick. But we'll get in, speaking of kids, right? But we'll get into polio towards the end. But yes, it's good to be not a bubble person. My sister's a bubble person, and I try to like, as soon as your kids come over, I'm like, take your shoes off, like, take that sweater off, like, oh my gosh, she like bundles them up, and I'm like, no.
I'm the floor.
She's like, oh, like they can't be like without their socks, and I'm like, no, socks off. So yeah, or they're like, oh, I have to fight with my family a little bit sometimes. I'm like, no, that's not good for them. And then guess what? They get sick all the time. I'm like, see, there was a video of my parents have a video of me like eating sand when I was like 1. I'm like, I never get sick. So there you go, it was sand. There you go, I knew it, I got it. Okay.
Which is what I did too is latex. I used to be really allergic to it, but then I used to always touch balloons as a kid, so now I'm not that allergic to it.
There you go. There you go. Don't be a bubble. Okay. What are some advantages or disadvantages of having an envelope? Okay. Advantage, when you have an envelope, you're kind of hiding, because that membrane that the envelope that the virus has, it gets its membrane from a cell as it exits. So it's your membrane that it's covered in. So is it harder for your immune system to detect? Yeah. So it's kind of like cloak of invisibility, right? It's hiding itself within having that coat of membrane that is your cell, okay? Also, that envelope helps fuse with your cells, because the membrane is lipid, your cells are lipid. So all they have to do is touch, boom, fuse together. Ta-da, super easy. Okay, fusion. That's how your virus will get inside. It fuses, takes its coat off, and then there goes the DNA, okay? Disadvantage for envelope is that it is easier to kill, right? It's more susceptible to a lipid solvent, right? Something like just soap, okay? A degermer. More sensitive to antimicrobial agents. There's a narrow pH and narrow temperature range because it's a lipid, right? So advantages, disadvantages to having an envelope. Okay. Some functions. Okay. So weird. Okay, anyways, so protein code, right? If you don't have an enveloped virus, then it's just a capsid, okay? It's another layer of protection. So like your capsid is protecting it, and then you have a membrane. So it's like double protection. You can also have you can evade certain mutations, right? Other pretty cool features to having an envelope or not having an envelope.
Are the capsids considered the nakeds?
Yes, if you only have capsid, then you're naked. If you have an envelope or a membrane, then you're an enveloped virus. Okay. The capsid can come in different shapes. So you can have a helical virus. So tobacco mosaic virus, that was an example of a helical virus. You have your polyhedrovirus, which was that adenovirus that we saw earlier. HPV is also a polyhedrovirus. And then a complex virus, aka bacteriophage. it looks a little complicated, okay? On top of the shape of the capsid, you can also have a membrane or no membrane. Okay? So we'll get some examples. So at the top, okay, this is tobacco mosaic virus, okay? It's arranged in a helix, and then there's no membrane. That's it. This one, you're looking at influenza. Influenza is a helical virus, and then it has a membrane over it. Okay? So both can exist. Okay? Polyhedrovirus. So adenovirus is an example. It's an isohedral, so it has 20 faces. Do you need to know 20 faces? No, I just care many sites, right? Polyhedral, poly means many, many sites. Not even like count the sides. It's a bit much, okay? Yeah. Poliovirus is also a type of polyhedral shaped virus. Okay? Okay, and then complex virus is just bacteriophages, right? They kind of just look like an alien. So they have a couple more structures, right? So they still have, right, genetic material. They have the capsid, but then they have some extra components, okay? They have a sheath, and then they have a tail fiber, and a pin, and a base plate. And this is what's gonna help it. Look at this microscope picture. That, you see, that's the alien. Okay, that looks crazy. Well, I mean, it looks like the alien from like Chicken Little, right? So they latch on to the cell membrane, and then they use the base plate, and they inject their DNA straight into the cell. Isn't that crazy? Like it just latches on, it's like, and it's like, and then it just lets the DNA straight in there. Okay. So complex is just bacteriophage. It looks fancy. It has a bunch of other components to it. Okay. You can also have poxviruses. So poxviruses, they don't have a capsid, right? This is the one that doesn't fit. So there's no capsid. It just has multiple membranes that surrounds nucleic acid. So that's our exception to the rule. Smallpox is an example. We'll get all into smallpox later. Okay. And then host range. So here's where we talk about ligand, right, receptor interaction. So here you have just a capsid, right? So this would be a naked virus. Okay. This protein, right, the capsomere, is being recognized by one of your receptors, okay? Here, we have an enveloped virus. So we have membrane and then spikes. So this spike is being recognized by one of your receptors. 8, okay? So there is a specific interaction. So the purpose is you have to be able to recognize the virus for the virus to come inside your cell. Okay. How we name viruses, different. We name them based on, are they DNA or RNA? And then we categorize them based on how they replicate and their morphology, right? We just mentioned how they're shaped. Depending on how the capsomere is shaped is what type of virus it is. We can also group them by, we call them families, right? So if you belong to the family Viridae, you add Viridae at the end. So in this example, you have Retroviridae. Okay. Genus, they add virus at the end. Okay. For example, human immunodeficiency virus. So that's telling you this is a virus that affects humans and it's causing a deficiency in your immune cells. So it's in the name, right? Very different from our binomial, okay? Then you can also add 1 or 2 to dictate a different strain, right? So you can have HIV-1, right? Or you have like RSV-1 or like RSV-2, right? That just says different strain, okay? So it's different way, right? Not binomial. Okay. So, 6:30. Okay, I'm gonna stop here so I can give you your quizzes. Okay, anyone else? No one else is presenting, right? Like paranoid. Let me give you your quiz. Okay, make sure you give it back. Yes.
She wants to present.
Oh, okay, present, and then we'll do quiz.